So we're still in gram-positive cocci, but this time we're discussing the other papillars gram-positive cocci, and that would be streptococci. So they are in this part of the schema. So as what I did mention to you a while ago, streptococci are catalase negative, and usually we differentiate them according to their characteristics in blood agar plate, if they're alpha hemolytic, they're beta hemolytic, or gamma hemolytic. And they don't grow in mannitol salt agar, again because of the fact that it's inhibitory because of the presence of 10% sodium chloride. So streptococci are generally gram-positive cocci in chain. They don't have flagella, they don't have spores, they don't have capsule, except for one species, that would be Streptococcus pneumoniae. And they are facultative anaerobic, and they require carbon dioxide for growth. So they are said to be what? Capnophilic. They're capnophilic. In blood agar plate, they produce pinpoint hemolysis, and there are about 29 recognized species of Streptococcus. Okay, so this would be the gram-positive cocci in chain. So streptococci. And how do we classify them? We classify them according to academic or Virgis classification, Smith and Brown classification, Lansfield classification. Okay. Academic or Virgis classification depends on the temperature. Okay, it depends on temperature requirement. So the pyogenic group cannot grow at 45 and 10 degrees. So an example would be Streptococcus pyogenes. They are capable of producing past, hence the term pyogenic. And then the Viridans group can grow at 45 degrees, but they cannot grow at 10 degrees centigrade. The Viridans group is the only group that doesn't have the M protein. So no M protein. So that's why later on, they cannot be classified under the Lansfield classification. Anyhow, the Viridans group includes Streptococcus mutans and salivarius. So they are found in the oral cavity. Strep mutans can cause dental caries. The most commonly isolated bacteria for dental caries. Enterococcus group obviously is isolated from the gastrointestinal tract. So they can grow both at 45 and 10 degrees. Streptococcus fecalis is an example. Lactic group. These are the lactic acid producing bacteria, Streptococci. So they can grow at 10 but not at 45 degrees. So representative organisms would be Streptococcus lacticola. Smith and Brown classification is based on blood agar plate, hemolysis in blood agar plate. So we have alpha, beta, and gamma. Strep pneumoniae is an example of the alpha hemolytic streptococci. Strep pyogenes is beta hemolytic. Gamma streptococcus would be Streptococcus faecium. The Rebecca Lansfield classification is based on the presence of specific carbohydrate antigen known as the C polysaccharide. So the virulence factor would be the M protein. I told you a while ago that the Viridans group do not have M protein. So since they don't have M protein, then they cannot be classified under the Lansfield classification. So in the Lansfield classification, we classify them as Group A, Group B, Group C, and Group D. So all of them, all strep, could be classified under here, except for the Viridans group. Group A streptococci are very pathogenic. This group is very pathogenic to humans. They cause the so-called post-streptococcal infections. What do we mean by post? Post means after. Usually as a complication of having streptococcal infection. So after a sore throat, let's say a sore throat is caused by strep pyogenes, you can have complications. And one of the complications would be the acute rheumatic fever, and the other one would be the acute glomerulonephritis. That's why you don't ignore simple sore throat, because this may result into streptococcal, post-streptococcal infections, which could be more dangerous. The complications due to streptococcal infections are actually more dangerous than the actual infection itself. So, this could affect your heart and your kidneys. Scarlet fever, pyoderma, tonsillitis, endocarditis are also associated with Streptococcus. Group A, and the most commonly isolated group A is Streptococcus pyogenes. So group A is not usually considered as part of normal human flora, as it is the strain of strep that is most oftenly associated with the disease. So the most common site where you can isolate group A would be the pharynx, particularly here at the nasopharynx. So aside from that, aside from this place, we can isolate group A from the nasal vestibule, nasal entrance, okay, nasopharynx, epiglottis, tracheal larynx, even here at the bronchioles. So most of the time we do throat swab in trying to isolate, and if you have tonsillitis, most of the time you'll be able to see white inflammatory plaques in a swollen tonsil. So if you have tonsillitis, which by the way every month I have it, so you find it difficult to swallow, that could also be accompanied by fever. And erysipelas is another one. Erysipelas is also due to Streptococcus infection. Scarlet fever is caused by strep pyogenes, particularly the scarlatinae. It is also known as the pink tongue or the strawberry tongue. One of the tests that you can use is the Dick's test. I can see a smile on your face, but you're not using any part of the body but your arm. Not the dick. I'm being recorded. Okay, you're using an arm. Your test arm and the control arm where you place toxoid. The test arm, you place toxin. Okay? You inject toxin. So, if there's an erythema, it means that you're exposed to the organism. So, another one is the Schultz-Charlton phenomenon. Why is it important? Schultz-Charlton phenomenon is also known as the blanching phenomenon. If a person's skin is red already, there are many reasons why the skin has rashes. It could be caused by parvovirus, the B19 virus, the fifth disease. It could be caused by, there are actually 5 bacteria that could cause skin rashes. If you want to know if the skin rash, or not really 5 bacteria, but 5 organisms that could usually cause skin rashes. If you want to know whether the skin rash is due to strep pyogenes or scarlet fever, you do the Schultz-Charlton phenomenon. So, if after giving toxoid and then the rashes disappear, then that that particular skin rashes is due to scarlet fever. Not really toxoid, but you inject the anti-erythrogenic toxin, because the toxin that is responsible for the scarlet fever is known as the erythrogenic toxin. Yeah, this is the pink tang. Okay, due to scarlet fever. Okay, to test group A, we can also do the bacitracin test. A test that will allow us to differentiate beta hemolytic streptococci from non-group A beta hemolytic streptococci, because if the streptococci can cause beta hemolysis, it doesn't necessarily mean that it is due to group A. There are other groups of streptococci that can cause beta hemolysis. To differentiate them, then you have to use the bacitracin test. Use taxo A, which is, by the way, made up of 104 unit of bacitracin, and a positive result is characterized by the zone of inhibition. So if there is a zone of inhibition here, okay, as you can see, there is a zone of inhibition, then this one is considered to be as positive for bacitracin test. Okay, so no inhibition means it's negative. Okay, so the PYR test, the principle is hydrolysis of L-pyrrolidose. Okay, hydrolysis of L-pyrrolidose. Okay, naftilamide by group A beta hemolytic streptococci. So a positive reaction is the presence of cherry red color. Group B. So we have discussed group A already. Then we have group B streptococci. Representative organisms under the group B is Streptococcus agalacti. It's a normal flora of the genitourinary tract of female, and it's normal for a female but not for the infant, not for the neonates. That's why during childbirth, Strep agalacti could cause neonatal sepsis, neonatal meningitis, and post-puerperal fever or fever after the child has been delivered. The organisms could rapidly hydrolyze sodium hippurate, and the test that is being used for determination of group B is known as the CAMP test, an acronym that stands for Christie, Atsky, Munch, Peterson. The group B streptococci is found at female genital urinary tract. So these are the parts where we can find the group B. So if the child, okay, is born, so that child has to pass through the area where we can find Strep agalacti, and that's how they get neonatal sepsis or neonatal meningitis. So here's an example of a child that died from neonatal sepsis. Here's a CAMP test. So what you have here is an arrowhead hemolysis. Colonies of Staph aureus, colonies of Strep agalacti, when they meet in the middle, they will produce hemolysis that's shaped like an arrow. So that's arrowhead hemolysis. Positive reaction for the CAMP test. Group C streptococci mostly are animal pathogens. In humans, representative organism is Equisimilis, but in horses, these are the equi. Most of the time, they infect the upper respiratory tract. All of them are beta hemolytic except for Streptococcus agalacti. Group D could be divided into two groups, the enterococcal and the non-enterococcal. Enterococcal obviously means that these are isolated from the gastrointestinal tract. Non-enterococcal would be isolated from other parts of the body aside from the gastrointestinal tract. to differentiate them, okay, we use sodium chloride and penicillin. So, enterococcal group can grows at 6.5% sodium chloride, which means they could tolerate salt, but they are not killed by penicillin. They are only inhibited, but not necessarily killed by penicillin. Unlike the non-enterococcal group, they are inhibited by the salt, and at the same time, they are killed by penicillin. So this one could cause UTI, enterococcal group can cause UTI, cardiovascular infection, and meningitis. Examples are Fecalis, Fisium, and Durans. While non-enterococcal, examples would be Streptococcus bovis and Equinus. This can cause UTI and endocarditis. So remember, penicillin and salt to differentiate group D into enterococcal and non-enterococcal. Okay, so what are the different virulence factors that could be caused by Streptococcus? One of them is streptokinase, an enzyme that could lyse coagulated plasma. The same principle as staphylokinase. Streptodornase could destroy the DNA. Erythrogenic toxin could lyse RBC. Hyaluronidase, I think I did mention this a while ago, spreading factor that could destroy hyaluronic acid in the connective tissue. Hemolysin could lyse RBC, but if it's from Streptococcus, we specifically called it streptolysin. Okay, speaking of streptolysin, okay, do you know that there are two types of streptolysin? Streptolysin S and the other one is streptolysin O. Okay. Streptolysin S is actually oxygen stable. That's why they are capable or responsible for surface hemolysis. Unlike streptolysin O, which is oxygen labile, that's why they are responsible for subsurface hemolysis, because at the subsurface, this one is not exposed to oxygen. So streptolysin O is important for subsurface hemolysis. Streptolysin S is non-antigenic. Unlike streptolysin O, it is antigenic. Therefore, there's no such thing as anti-streptolysin S, because this one is not antigenic. Unlike ASO, there is a such test known as ASO, or the anti-streptolysin O. ASO or anti-streptolysin O is a serological test to detect the presence of antibodies against streptolysin O. Usually, if you've been infected with Streptococcus, you produce antibodies against streptolysin O, and you call that antibodies as ASO. So how do you perform ASO? Usually, we extract blood from the patient and get its serum, and then we add 1 drop of serum into the slide, and then 1 drop of latex reagent. We mix it and look for the presence of agglutination. If agglutination is present, then it's positive. What does it mean? It means that you have been infected with Streptococcus. And what is the implication? If your ASO titer is very high, because the normal range of ASO is supposed to be less than 200 international unit per ml. If your range is beyond 200 international unit per ml, there's a big chance that you'll get post-streptococcal infections. And what are the examples of post-streptococcal infections? Rheumatic heart disease or acute rheumatic heart fever and acute glomerulonephritis. Okay? So that's why in the hospital, there might be a request in the hospital and the request says ASO. Oh, the doctor is requesting for my dog. No. ASO stands for anti-streptodizin O. Okay. Now, the Viridans streptococci. What did I tell you about the Viridans streptococci? Yes. They don't have M protein. But examples are strep mutans. Examples are strep mutans, strep sanguis. They are able to form biofilms on tooth surface. Again, biofilm. And they are capable of causing plaque. Okay, plaque. So, how? Why they are important? Let's say this is a tooth. And here, you can find the nerves. Okay, this is the enamel. Enamel, this is the then after that, the dentine, this would be the pulp. So, if you have plaque on the surface, you have bacteria here, they will ferment carbohydrates because of the food debris, because of the tinga. They're not flossing, they're not mouth washing with antiseptics. So, if they ferment the carbohydrates, again, what did I say about the product of fermentation? Acid. And acid destroys the enamel until such time that the plaque has reached the pulp. And once it has reached the pulp, that's the time when you feel pain. Now have tooth decay. So most of the time, before you reach the decay reaches the pulp, so we have restorative procedures. If you go to dentist, they will add pasta here. And diba? So that if you remove it, if there's a pasta, you prevent further destruction. Okay. Do you know that if you have dental caries, not caries, dental caries, dental caries. If you have dental caries, there's a big chance that you'll get heart disease. Okay. Why? Because the organism, okay, these organisms are low pathogenic potential, but even if they are low pathogenic potential, from the oral cavity, they might go into your circulatory system, goes to your heart, and destroy the inner wall of the heart, known as the endocardium. Once it is destroyed, you develop a disease known as endocarditis. Which means that a healthy oral cavity would also mean healthy heart. This is an example of infective endocarditis on the aorta. It's the major blood vessel of the heart. So this is the aorta. Streptococcus pneumoniae is another example of gram-positive diplococci, quite unique because of its characteristic lancet shape, and they are encapsulated. Could cause lobar pneumonia, wherein a person would have a characteristic of rusty colored sputum. So the sputum appears to be rusty. This could also cause meningitis and septicemia and otitis media, or the luga, or the infection of the middle ear. They are facultative anaerobe and they are capnophilic, which means that they require carbon dioxide for growth. Alpha hemolytic, they have a dome-shaped colonies in bladder plate. They produce the so-called SSS, 3S only, not the 4S. So it means specific soluble substances and the soluble antigen. The test is known as the Francis test. Dick's test for scarlet fever, Francis test for strep pneumoniae. Okay, so the organisms are encapsulated. So here you can see the capsule. Lancet-shaped diplococci. When you say diplococci, they appear to be in pairs. Lancet-shaped diplococci. So only those strains of strep pneumoniae which contain capsule can cause diseases. If the strain doesn't have capsule, then they are non-pathogenic. Okay? So, the disease is known as Streptococcus pneumoniae. So, the organism is known as Streptococcus pneumoniae, and they can cause respiratory tract infection. Streptococcus pneumoniae is one of the most common organisms which would cause otitis media, or the infection of the middle ear. At the lower respiratory tract, they could cause pneumonia. It could also be invasive, causing bacteremia and meningitis. So here's lobar pneumonia. Okay. And then, how do we differentiate pneumococci? Pneumococci, by the way, is the common name of strep pneumoniae. We can have the biosolubility test. Strep pneumoniae is biosoluble. Other streptococci cannot be dissolved in bile. Strep pneumoniae can ferment the sugar inulin, while other strep cannot ferment it. Neufeld-Quelling test is actually known as the capsular swelling test. Here, we add antibodies plus methylene blue as the stain for capsule, and then we check under the microscope. If the capsule is swollen, then that's positive for strep pneumoniae. Unlike other strep, since they don't have capsule, so nothing will swell. And then quinidine. Sa antibiotic quinidine, pneumococci are susceptible to it, but other strep are not. They're resistant. Okay, optochin, one of the most popular tests for pneumococci. Again, pneumococci are sensitive, other strep are not. The mouse virulence test, later on I'll show you how they discovered the strep pneumoniae. So, mouse dies within 16 to 48 hours, unlike in other strep, remains alive. So, in mouse virulence test, here's the optochin. Here's the optochin test. A test that will differentiate strep pneumoniae from other strep. Unlike in bacitracin, you're using Taxo A. Here, you use Taxo P, which is made up of ethylene, hydroxyprene, and a passive reaction is characterized by the zone of inhibition. So here's an example of a positive optochin test. Here's an example of a negative optochin test. See you on Wednesday.
Genetic information between organisms in the same generation is called horizontal gene transfer. 3 types of horizontal gene transfer occur in bacteria: transformation, transduction, and conjugation. All three mechanisms involve a donor cell that transfers a portion of its DNA to a recipient cell, which may incorporate the new DNA into its own, becoming a recombinant organism.
Okay, so in this particular information, we have already discussed conjugation. Remember? Conjugation is done by means of what? By means of villi. Very good. And then, vertical gene transfer, the bacteria, let's say during binary fission, the genes are being transferred to its progeny, its daughter cells, its progeny. Unlike horizontal means that the organisms are being transferred into another bacterial cell. And this explains why some bacteria will have capsule, some bacteria will not have any capsule. And how did they discover it? One of the experiment deals with Streptococcus pneumoniae. We call it the Griffith experiment. And the Griffith experiment is an example of transformation. Here's what happened. In the Griffith experiment, Griffith performed two strains of the bacteria, the R strain and the S strain. As you can see, the R strain doesn't have capsule, while the live strain would have a capsule. And then the third one is the S strain hit kill, which kills the bacteria. And the other one would be combination of the R live. R live means no capsule, and S the hit kill. So here's what happened.
The unencapsulated cells of the rough or R strain of Streptococcus pneumoniae do not cause disease. When mice are injected with these cells, their immune system efficiently deals with this invader and the mice live.
So the mouse survive. So it is not caused by Streptococcus pneumoniae. Okay. How about the other one?
Cells of the smooth or S strain of Streptococcus pneumoniae produce a capsule that allows them to escape phagocytosis by the body's defensive white blood cells. When mice are injected with these cells, the mice die.
Okay. Capsule, within 16 to 48 hours, the mouse dies. Because the capsule prevents phagocytosis. So this allows Streptococcus pneumoniae to exhibit its virulence. How about the S strain hit kill?
Heat can be used to kill the smooth strain of Streptococcus pneumoniae. When mice are injected with these heat-killed cells, the mice live.
Okay, so the mouse lives because you remove the microorganism, but the capsule remains. The fourth one is the R live plus the heat kill. Look, this is interesting.
Surprisingly, when mice are injected with a mixture of harmless, heat-killed, smooth-strain cells and harmless, live, rough-strain cells of Streptococcus pneumoniae, the mice die. Live, smooth-strain cells can be found in the blood of these dead mice. The harmless, rough-strain cells must have gained the genes to produce capsules, like those of the virulent, smooth-strain cells.
So that's transformation. You kill the organism, but the gene remains. So those bacteria without the gene will get the gene. Eventually, what will happen? They become encapsulated. Which means, for example, I am the R live and he is the S live, S strain live. He died. But, sorry, JM. He died, but his clothes remain there. So I got his clothes. So I now have his clothes, and I'm now a virulent strain, and I will cause the death. Di ba? So the gene transferred, I'm not his son, so I'm not a progeny. So it transferred from one bacteria horizontally to another. So that's how Streptococcus pneumoniae will get it.
Some bacteria are capable of taking up fragments of DNA from their surroundings and integrating the fragments into their own chromosomes by recombination. Cells capable of taking up DNA from their environment are said to be competent. Only a few genera of bacteria are naturally competent, such as the S. pneumoniae in this example, but other bacteria, including E. coli, can be made competent through simple laboratory treatments.
A while ago, we also discussed how Staphylococcus becomes pathogenic, becomes virulent by having a phage treatment, because the phage or bacteriophage is a virus that could deliver the genes. So if this is transformation, the other one is called transduction. So here's what will happen in transduction.
In transduction, DNA from the chromosome of one cell is transferred to another cell via a replicating virus. A virus that infects bacteria is called a bacteriophage or phage.
See? So when a bacteriophage infects the bacteria, bacteriophage is a Latin term which means bacteria eating virus. So the virus will not literally eat, but will cause lysis. There are two types of life cycle: lytic cycle, and the other one is lysogeny. In lytic cycle, as what happened into the first bacteria, the bacteria lyses. The other one is lysogenic cycle. When the bacteriophage infected the bacteria, the gene coming from the previous bacteria could be inserted into the recipient cell. So that's what you call transduction. So here's the donor.
Of generalized transduction in bacteria, a bacteriophage attaches to a bacterial cell wall and inserts its DNA. The phage commandeers the bacterial cellular machinery to synthesize new phage DNA and produce phage proteins. In order to completely convert the bacterium into an efficient virus-producing factory, some of the phage proteins break the bacterial DNA up into small pieces. Normally, phage DNA is coated with phage protein to form new virus particles, but sometimes pieces of bacterial DNA are mistakenly packaged in phage proteins. These sloppily packaged phage particles carry bacterial DNA rather than phage DNA and are called transducing phages.
So the transducing phage will now infect another bacteria.
When a transducing phage, composed of phage proteins and bacterial DNA, infects a new host, the phage injects the bacterial DNA into the new bacterial host. The infected bacteria sometimes incorporates this DNA into its own chromosome through recombination, resulting in the transfer of genes from the donor bacterium to the recipient bacterium.
So the principle of transduction is very important. Later on, we will be discussing Corynebacterium diphtheriae. We will be discussing Vibrio cholerae. The reason why this bacteria becomes virulent because of the genes. And these genes will make them virulent, and not all bacteria, let's say not all Corynebacterium diphtheriae will have the gene. They will have the gene if they have been infected with a phage. Not all Staph aureus, which is our previous lesson, would have the gene that could manufacture enterotoxin. They could only have the gene if they have been infected with a phage. But the requirement is that that phage should become a transduced phage, which means that they had previously infected another bacteria that contains that particular gene. Okay? So these are two very important lessons in horizontal gene transfer, which is of course related to our lesson in Streptococcus pneumoniae and in previous discussion that we had a while ago.